It should come as no surprise that purine and pyrimidine nucleotides are the building blocks of DNA. Without them, we can't make DNA, and if we can't make DNA, then life would cease to exist. That includes you too, rapidly dividing cancer cells and lymphocytes. To understand how purine and pyrimidine nucleotides are made, we shall go back to a simpler time. The setting is Paris. The year is 1482, and the Notre Dame Cathedral was the most celebrated place in all the land, not to mention free of any fire-related damages. The cathedral was home to an innocent hunchback who lived in the bell tower. The hunchback was informally adopted by the evil and manipulative high priest Claude Froyot. Atop the roof, we're going to illustrate the steps of De-Novo purine and pyrimidine nucleotide synthesis among the gargoyles and statues of the Notre Dame Cathedral. And just as a heads up, the gutters on the roofs connecting each molecule represent the actual synthetic pathways. Recall that nucleotides are composed of a nitrogenous base, ribose or deoxyribose sugar, and one or more phosphate groups. The nitrogenous bases include the purines, adenine and guanine, and the pyrimidines, cytosine, thymine, and uracil. Remember that thymine is only present in DNA, and uracil replaces thymine in RNA. Let's first go over the products of purine and pyrimidine nucleotide synthesis. These pure as gold statues containing two ringed halos represent the purine nucleotides, adenosine monophosphate, or AMP, and guanosine monophosphate, or GMP. The statue on the left is holding a lamp. The last three letters will help you remember AMP. The statue on the right looks grumpy, a grump for GMP. Now onto the pyrimidine nucleotides, uridine monophosphate, UMP, cytosine triphosphate, CTP, and deoxythymidine monophosphate, DTMP. They'll be represented by three different gargoyles, each having a single hexagon halo. A unicorn imp for UMP, a cat with three P's for CTP, and a toad with a missing eyeball for DTMP. Why no eyeball, you ask? Because it's deoxythymidine, so it's missing an eyeball, or as we call them here at Sketchy, an O-ball. It's missing an oxygen, okay? Deal with it. Anyway, I know that the story about that toad was absolutely riveting, but I'm sure you're itching to find out how we make these nucleotides. Both purine and pyrimidine nucleotides are made from ribose 5-phosphate, represented by this pentagon and P atop the spire. Please watch our pentose phosphate pathway sketch to learn how ribose 5-phosphate is made if you haven't already. First, PRPP synthetase transfers a pyrophosphate group from ATP to ribose 5-phosphate, making phosphoribosal pyrophosphate, or PRPP. Below the spire, a worker is using an ATP power drill to attach a P staff to the pentagon-shaped pedestal and double P staff. He's PRPP synthetase, and he's making PRPP. Yeah, we know there weren't battery power drills back then, but this is sketchy, and anything is possible. We've reached our first juncture as PRPP can be used to make either purine or pyrimidine nucleotides. We'll begin with the purines. PRPP will undergo several biochemical reactions to eventually make inosine monophosphate, or IMP, depicted by the imp gargoyle. We won't talk about each reaction in detail because we don't want to make you gag, but you should know that glutamine, aspartate, and glycine are needed to make IMP. We also need tetrahydrofolate, or THF. Well, looks like somebody's already got us covered. This imp gag oil, if I do say so myself. Behold, the evil high priest Claude Froyot has captured the beautiful azamerelda. The captured azamerelda will help you remember 6-mercaptopurine, or 6-MP, and azathioprine, a prodrug of 6-MP. They block de-novo purine synthesis by inhibiting the conversion of PRPP to IMP. That's why we show azamerelda kicking the imp gargoyle. 6-MP and azathioprine treat certain cancers and autoimmune disease. Cancer cells proliferate rapidly and need purines to replicate their DNA. So what better way to slow cancer cell replication than to block purine synthesis? In autoimmune disease, the goal of treatment is to prevent leukocytes from attacking the body's own cells. The problem is, anytime we block purine synthesis, other normal rapidly dividing cells are affected too, and that's where all the bad side effects come from. Please check out our Sketchy Farm course to learn more about these drugs. In the next two steps, IMP will eventually be converted to AMP. First, adenylosuccinate synthetase uses GTP to turn IMP into adenylosuccinate. This step requires aspartate. Then, adenylosuccinate lyase cleaves adenylosuccinate into AMP and fumarate, which enters the TCA cycle. IMP is also a precursor to GMP, and again, this requires 2 steps. First, IMP dehydrogenase oxidizes IMP to XMP, or xanthine monophosphate, while producing NADH. Then, GMP synthetase uses ATP and glutamine to synthesize GMP. Let's talk about the drugs that block GMP synthesis: mycophenolate mofetil and ribavirin. They inhibit IMP dehydrogenase. Illustrated here is quasi-mofetil accidentally kicking the grumpy gold statue while desperately trying to save Esmeralda. To remember mycophenolate mofetil, think quasi-mofetil. Notice his ribs are exposed, ribs for ribavirin, and him kicking the grumpy statue will help you recall that these drugs block GMP synthesis. Okay, let's move on and discuss how the pyrimidine nucleotides, that's UMP, CTP, and deoxy-TMP, are made. First, carbamoyl phosphate synthetase 2, or CPS2, synthesizes carbamoyl phosphate from glutamine and CO2. This step requires 2 ATP and takes place in the cytosol. Let's turn your attention to the worker in a 2 shirt using an ATP-powered drill to make an oil lamp. He's CPS2. Don't confuse him with CPS1, which is found in the mitochondria and is a part of the urea cycle. Please check out our Urea Ungle Crew sketch to learn more. As we mentioned earlier, CPS2 requires glutamine and CO2, and can be remembered by the G-wick and black smoke. The product is carbamoyl phosphate, or for our purposes here, an oil lamp. In the next 3 steps, carbamoyl phosphate eventually gets converted to orotic acid, represented by the otter gargoyle. While you don't need to memorize each step, you should know that making orotic acid requires aspartate. You'll see the letter A incorporated into the tower behind the otter gargoyle. The A stands for aspartate. The drug luflunomide, used to treat rheumatoid arthritis and psoriatic arthritis, blocks orotic acid synthesis by inhibiting dihydroorate dehydrogenase. This enzyme catalyzes the last of the 3 steps needed to convert carbamoyl phosphate to orotic acid. Therefore, we wouldn't be able to make pyrimidine nucleotides. Illustrated here is Le Fleu, a French bird, pooping on the otter gargoyle. Oui, Le Flunomide, though he should probably be called Le Poo. In the next step, orotic acid combines with PRPP to make UMP via UMP synthase. The worker chiseling the unicorn imp gargoyle is UMP synthase. So what happens when UMP synthase doesn't work? As you can probably infer, UMP synthase deficiency results in accumulation of orotic acid because it can't turn into UMP. The falling chiseler and everything immediately surrounding him will illustrate what you need to know regarding UMP synthase deficiency. Excess orotic acid leaks into the urine, causing orotic aciduria, represented by the yellow substance of unknown origin leaking from the otter gargoyle. If you've watched some of our other sketches, you'll know that the red firework blast represents megaloblastic anemia. This occurs because of disruption of DNA replication inside red blood cells. Just a side note, orotic aciduria also occurs in ornithine transcarbamylase, or OTC, deficiency, a urea cycle disease. However, those patients won't have megaloblastic anemia because pyrimidine nucleotide synthesis isn't affected. Instead, they'll have hyperammonemia because they can't make urea from ammonia. In the next step, UMP is converted to UDP. We've shown another unicorn gargoyle, but this one has a double P staff. UDP eventually becomes CTP. UDP may also be converted to deoxy-UDP or DUDP. Ribonucleotide reductase catalyzes this step. Let this RNR sign remind you of ribonucleotide reductase. There's also another unicorn gargoyle, but this one's round eyeball is falling off. It can't see because it's deoxy-UDP. Hydroxyurea, used to treat myeloproliferative disease, chronic myelogenous leukemia, and sickle cell anemia, inhibits ribonucleotide reductase. We drew a hydro rock area to show hydroxyurea. Note the water splashing the R and R sign because hydroxyurea inhibits ribonucleotide reductase. In the next step, DUDP is converted to DUMP. The unicorn gargoyle with a missing eyeball and 1 P staff is DUMP. After making DUMP, bimidylate synthase uses 5,10 methylene tetrahydrofolate, MTHF, to turn DUMP into DTMP and dihydrofolate, DHF. The worker chiseling the toad imp gargoyle is thymidylate synthase. The group of 4 leaves containing the number 5 and 10 branches in the circular water fountain is 5,10-MTHF. 5,10-MTHF is a derivative of THF, the active form of folate. THF derivatives transport single carbon groups during metabolic reactions. In this particular reaction, 5,10-MTHF is converted to DHF. The enzyme dihydrofolate reductase makes sure that there's an abundant supply of THF so we can continue making purine and pyrimidine nucleotides. It reduces DHF into THF. The pair of leaves is DHF and the other group of 4 leaves is THF. The drugs trimethoprim, methotrexate, and pyrimethamine inhibit dihydrofolate reductase. Note they all have meth in their names. Trimethoprim can be remembered by the Try-Me bag, and the Pyre logo is pyrimethamine. Finally, that worker has some methotrexate meat sticks.
Part of this complete breakfast may cause substantial side effects including diarrhea, alopecia, thrombocytopenia, and thrombosis. For display purposes only, not part of any appropriate breakfast. Enlarge to show texture.
Let's conclude this sketch by also mentioning the drugs that inhibit thymidylate synthase, 5-FU and capecitabine. We showed 5-FU as a full guy motioning with his 5 fingers that he doesn't want any more kebabs. And that capsized boat is capecitabine. Okay, let's wrap up this sketch. Purine and pyrimidine nucleotides are made from ribose 5-phosphate. First, PRPP synthetase uses ATP to turn ribose 5-phosphate into PRPP. In order to make the purine nucleotides, PRPP undergoes several steps to make IMP, which eventually gets converted to either AMP or GMP. 6-MP and azathioprine block IMP synthesis. Mycophenolate mofetil and ribavirin block GMP synthesis. In order to make the pyrimidine nucleotides, CPS2 uses 2-ATP to make carbamoyl phosphate from glutamine and CO2. Next, carbamoyl phosphate eventually becomes orotic acid after combining with aspartate. Leflunomide blocks orotic acid synthesis. Then, UMP synthase makes UMP from carbamoyl phosphate and PRPP. UMP synthase deficiency causes orotic aciduria and megaloblastic anemia. In the next step, UMP is converted to UDP. UDP can become CTP or be converted to DUDP by ribonucleotide reductase. Hydroxyurea inhibits ribonucleotide reductase. Afterward, DUDP is converted to DUMP. Finally, thymidylate synthase turns DUMP and 5,10-MTHF into DTMP and DHF. Dihydrofolate reductase turns DHF into THF. Trimethoprim, methotrexate, and pyrimethamine inhibit dihydrofolate reductase, and 5-FU and capecitabine inhibit thymidylate synthase.